we have made series of questions what we ourselves got, and also most of our viewers, audiences have also requested for the certain technical aspects related to CAR-T, which they would love to understand.
Mm.
Uh, so we have compiled all those questionnaires, but I'll just start with a brief introduction. So I myself, Prashanth, so founder and director of Incepbio. I'm a microbiologist and having 18 plus years of experience in pharmaceutical domain. And we at Incepbio predominantly on provide services on engineering compliances like calibrations, validations, C&Q, and also turnkey projects. So that is where the CAR-T, which is the very happening topic today. Uh, in fact, globally, wherever I have traveled, so most of the new projects are focused on this. Though closely linked with biologics, it has its own challenges and other expertise requirement to
Mm.
ensure a success CAR-T project. So that is where we wanted to have Ramakanth on boarded and discuss on latest aspects of CAR-T and what are the pitfalls which people should avoid, and what are the latest technologies which can also people consider for overall scaling up of their CAR-T facilities. So with that, Ramakant, I request you to please introduce yourself to our audience.
Thank you, Prashant. Sure, I will try my level best to answer most of the questions with my knowledge and whatever I have experience in CAR-T and different other projects. So myself, Ramakanth, I have 14 years. I have done my master's MTech in biotechnology. Post to that, I was working for different companies like Biocon, Dr. Reddy's, Stellis, Strides, uh, also a couple of other companies like Hyde Engineering Consultancy and all those stuffs. So my key area is mainly on the process engineering side, designing and all those stuffs. So just to keep you on the CAR-T side, I have a little bit on experience on CAR-T because I am from biotech background. So a little bit more happy to be part of CAR-T because that's what my field was, like cell and gene therapy, editing genes and all those things that made me understand more and more into that. So good. Thanks for introducing Prashant for into this webinar.
Yeah. Thanks a lot, Ramakant. Also to all the viewers, Ramakant and myself were colleagues in Biocon almost 12, 13 years ago. Okay, we worked together on most of the monoclonal antibodies. And also, there is one small disclaimer which Ramakant was very keen on. So this is like his contribution to the entire community, and there are no financial aspects involved in this webinar. Okay. With that, Ramakant, is it okay? We'll start, discussing on the queries and questions what we have got.
Yes, Prashant, we can start that.
Okay. From your experience, Ramakant, what are the top three critical factors that determine a success of a CAR-T manufacturing facility? Just in a top level, what are the three main factors which people should always keep in mind?
Yes, so see, CAR-T facility is very new nowadays. Before I go to the critical factors, right, let's understand about CAR-T, what actually CAR-T is. I'll just take in 5 to 10 minutes so that everyone get
Yes,
an
please.
idea. So
Yeah.
this it's all comes under the umbrella of cell and gene therapy. In our biotech field, we used to do some gene editing and all those things and do some gel clotting and you isolate it, but never it has come to that phase that it is becoming helpful for a human being. So nowadays it is getting, so any cell and gene therapy which is good for human beings, especially editing the genes, editing the cell for the sake of good for the human being health, and that's for cell and gene therapy is. So under cell and gene therapy, right? So CAR-T, chimeric antigen receptor T cells. So many times you would have talk, right? Why CAR-T is used? Any idea, Prashant? Like
Mhm.
it's for cancer, right? So,
Yes.
so why why we are talking about CAR-T and cancer? Why not? There are a lot of maps, right? There are a lot of ADCs, antibody drug conjugates, which can cure uh, certain amount to the CAR-T cells, which can cure the cells. I think someone can mute. There is a disturbance. Thank you. So,
Um,
it's I can say that these CAR-T cells, why so important is, see, normally I will just tell you a small story about how it actually acts. See,
Please.
when a body is happily enjoying its regular health kind of thing, there are certain kinds of cells which can mutate, which can also act as a terror kind of thing, terrorist kind of thing. Okay, simple example. So they silently enter into your body, and no one knows when it enters, how it enters, and they silently enters. Once they enter into the body, they acts very similar to the good cells. So they are similar to our good cells. So
Okay.
they were escaping from our immune system, so our T cells cannot identify them. So when T cells are not getting identified, they are unable to kill the cancer cells or identify and kill them. So what will happen is they start growing. They take our food from these cells and they grow, multiply, and it becomes more and more. So that's where the cancer starts. So now, cancer
Okay.
can be treated by biological molecules also. Why CAR-T? So all these target on the cancer cells, but CAR-T is something, the T cells, which are the, I can say, army of our body,
Mhm.
which warriors of our body, which can kill the invade of any of the external organisms. So in this case, we are not, we are planning to modify the T cells. So you are making the army warriors more stronger so that it can naturally fight back on the cancer cells. So this is where the CAR-T comes up. So we are making our T cells, our own body T cells, more stronger, more target, and more easy to identify the cancer cells. And then what happens? It goes, it goes and hits once it triggers. So this is how it is differ from the other biological products, ADCs. They're more target on the cancer cells, but this we are making our own T cells more stronger. So this is the main thing on the CAR-T side. So coming to your question, what are the key, critical factors when we are talking about CAR-T facilities, right? So always
Yeah.
quality plays an important role uh, in the CAR-T. Uh, so it's a kind of quality by design,
Mhm.
how we are actually uh, setting up this facility for a CAR-T. Second thing is on what technology you are using, what methodology your product is developed. That's also an important stuff. The reason is nowadays it's emerging technology and no one has an experience how there are a couple of players in the market who has good experience, but everyone is learning as if they are into the manufacturing. So what kind of methodology you are using in the R&D and how easily it can be transferred to the manufacturing side. So that's
Okay.
also one critical factor. And especially in
Mhm.
CAR-T, right, logistics plays a very important role. The reason is CAR-T, especially, it has maybe autologous and allogenic, maybe different kinds of thing. Especially in autologous means where the patient blood cell is taken, modified, and given back to the patient. So in this case, like logistics, if the hospitals are very long, transportation, your cold
Mhm.
chains, how it works, there are a lot of factors which are involved in this. So already the cells are weakened because the patient is already weak and you
Yeah.
cannot mm, transport the T cells or the blood from the patient to the facility in a longer duration, because it has also a time to expand by
Yeah.
itself and all those stuffs. So I feel these are all the key factors with respect to critical factors for the CAR-T facility, Prashant.
No, I am in total agreement with you, and especially the last point what you were mentioning about, the one is the manufacturing and the administration of the overall, the processed drug to the patient in the defined estimated time is more critical here, and logistics plays a major role. And we're also doing a lot many CAR-T projects across India, and we almost find it very challenging to build these facilities because most of the times it is well within already existing and up and running facility.
Mm-hmm.
So it throws different challenge again, how to build facilities within Indian facility, because already there are patients, already there are not many other activities which are happening. So that itself throws a different challenge. But I totally agree with you, like the critical factors what people need to consider whenever they are putting their hands into the CAR-T facility. Okay. Also to our audience, if you have any questions, you can also type in the chat box. So we will ensure that the particular question will be asked to Ramakant and you will get the answer for that. Okay. Thank you. Uh, Sorry.
Pra
How does yeah, yeah. I'm coming again. Like, how does facility design directly impact the consistency and success of CAR-T manufacturing outcomes?
so facility design. So normally, uh, when we are talking about facility designs, uh, normally what will happen, right? You have a some kind of facility which is nowhere idea about CAR-T, and you build a facility. So that
Yeah.
makes you a different approach. So here I would seriously put into in this mainly on the process side. You need to understand on the process first. So on
Okay.
what technology it has been developed. For example, either it is developed, um, either it is PN 70 bag or G-Rex or Cocoon, what technology it has been developed. Based on that, the facility need to be designed. So it's like no need to build a facility where you have all closed process with an isolators. So this
Yeah.
becomes over designed. So
Okay.
first understand what's your product and how it is developed in your R&D, how the clinical trials are done. Based on that, you design the facilities. So again,
Okay.
here again it comes quality by design is first thing very important. And also especially the automation part. So better to go with good automation technology, because you are dealing with T cells which are last T cells of the patient, I can say,
Mhm.
because you're taking all the T cells from the patient and you're giving, modifying it, giving back to the patient. But already the patient doesn't have much T cells. You're taking the last T cells, so it should be very careful. And you need to return back to the patient for his survival. So that's the
Yeah.
reason quality by design, you need to consider good automations, you need to be having good man material flow, and also on the contamination controls, especially in the CAR-T. The reason is whatever the process you are doing in CAR-T is all, it's like I can say that completely sterile kind of thing, because there is no filtration process in this. At the end of the, before you give it to the humans, right, there is no any filtration. So
Yeah.
from the time of you take the blood or the T cells
Mhm.
from the patient and giving back to the patient, complete process should be a sterile kind of thing. It should be
Okay.
minimum bioburden. I can say no bio. That's the reason contamination controls are also plays a key role in facility design.
Okay. Okay. And also, just like one of the idea or thought which I keep saying is whenever we go for a new project discussion. So many a times people tell, no, it is just again one more lab, because when the facility is smaller, like especially when you go with smaller systems, it might be Cocoon or Prodigy. All it needs is basically the biosafety and basic connections. So many a times there is a notion, strong notion, that it is just like a lab. Why we need to consider it is like a proper GMP facility? Why we need to keep DCGI, CDSU, or USFDA in mind? So
Sorry, Prashant, you were breaking in between. You're talking about why.
it's like people are saying that okay, we have some network issues.
I think,
Is
Prashant,
it also the customers when they
I'm I'm unable to hear you, Prashant. Is to me only, or everyone has the same issue? Just an
Yeah, Prashant is breaking in between.
can you, yeah, can you see me now, Ramakant? Can you hear me now?
Now I can hear you.
Yeah. So what I was trying to tell here, Ramakant, like most of the times when we are visiting the customer, also there is a common notion that the entire facility is just one more lab, and we don't want to break our head in again keeping separate AHUs, and all these general talks are there, because most of the times, because these are done at different setups like hospitals and other things, though this becomes a common notion. So how do you think, this is this idea of taking it very lightly should not be done?
No, constructing a CAR-T facility or uh, in a compliance way, regulatory way, in a hospital is a positive note. There is nothing wrong in that. Only thing is how you control
Mhm.
your product in and out. But the thing is, what you need to understand is,
Yeah.
especially on the regulatory point of view. See, CAR-T, for example, you consider India. Okay, CAR-T is very new to India, so main thing is, if you need to get approval from regulatory, you need to educate a regulatory, because when a new technology comes, right, getting a regulatory document, or some kind of guidance, every member should be part of that, educate them and take it in a positive way,
Mhm.
and it should get fly. So first thing is, I feel like getting education to the regulatory bodies, if they are new to the CAR-T. Second thing is, you can consider the design very easy, very in a good manner in a hospitals also. It can be done in a separate facility also. But main thing is complying to the all the design aspects. You need to protect your product. So it doesn't say that I have a lab and I can manufacture the CAR-T with a minimum lab requirement. No,
Yeah.
it's at least a minimum requirement, having a dedicated HU, maintaining certain grades, you have proper man material flow, you have proper segregation procedures, you have certain testing procedures. All those things need to be taken care. If everything at the end, if you are able to take care of the product, that means you are good, your product is good.
Okay. Okay. Fine, Ramakant. Get it. Thank you. And see, moving on to the next question. So again, probably, we can throw more light here. What are the most common mistakes organizations make while setting up CAR-T GMP facilities?
uh, that's a good question. So it depends on different organizations. Okay, so
Okay.
uh, see, for example, some organizations, what they do is, they might not know about CAR-T facility, or they might not know about CAR-T, and they were willing to enter into that business. So in this case, you cannot implement their existing facility methodology into CAR-T. For
Mhm.
example, you have some fill finish experience, then
Yeah.
you cannot expect a fill finish uh, procedures and all those things in CAR-T. Or if you have an
Uh-huh.
upstream, downstream procedures, DS facility. So, right, um, people should be there for CAR-T because um, it's it's you're talking about the last T cell of the patient, so you need to be very careful when you're handling these things. So education is important and experience in CAR-T is very important. Properly training the team is very important, and also strategic planning, how to mm, build a new facility and all those things.
Okay. Okay. No, definitely we'll make a note of that, Ramakant. And moving on to the next question, probably you can give us more uh, clarity on this particular question, wherein how important is a process-driven design approach compared to traditional clean room design approach?
So that's what. So it's always important considering driven approach is a good approach always
Mhm.
in any of the, because if you have a traditional design and you try to fix that process in that uh, traditional design, you will end up maybe spending more. You will end up with maybe slow projects. You will end up lot of risks. So always, what I feel is process is very important. So based on your process, you design the facility. Not like every traditional facility has 5 rooms. You go ahead and build 5 rooms, and within that 5 rooms, let's build a CAR-T facility. No, that's
Okay.
that will that what happens there is, you know, you're losing money, you're losing time, you do a lot of modifications, and you might also do some compromise in your process because of the traditional approach. So my main thing is process driven facilities has a more
Hmm.
advantage.
Hmm. Okay. Okay, I get that point. So, in between Ramakant, so there is a one question from one of the audience here. So, the question here is from the person named BG. There's no full form, so I will take the initial BG. So, how critical is role of skilled manpower in CGT?
Yeah, so it's very important. Every step uh, in CGT should be done by experienced guys, because uh, you are actually handling with the T cells, and you need to give a good environment to the T cells when you are working with the CGT. Every step, I can say, like, from the FRCs to till you start your cell separations, or you activate it, or you transduct with vectors, viral vectors and expansion, and then following formulation. Every step you need an experienced person for the CGT. Regular guys,
Okay,
I'm not sure, but you need to understand each and every step of the cell and gene therapy.
okay. So I agree with you, Ramakant. Even our experience has been, in almost all the projects, wherein the person who come from that background and their skills matter the most for the success of the project. Uh, though smaller facilities might need just 2, 3 people to do the entire solutioning initially, considering the clinical trial phase and that timeline. But we also strongly believe that the right fit of the person with the right skills for the technology, what they want to deliver it to their patients, is very important for ensuring the project success. So I hope BG, your question has been answered. And
Uh,
I'll move on to the Ramakant. Please go ahead.
I would like to add on here also. I would also like to
Mm.
tell, if you use unskilled, not unskilled, experienced
Yeah.
guys who are industrial experienced guys, but doesn't have a CAR-T experience. So what will happen in this case? For example, you have like 15-20 years of experience in fill finish, for example, and they're very much happy with the isolator technology. And my
Yeah.
CAR-T is completely closed process with a Prodigy or a Cocoon uh,
Mm-hmm.
or a G-Rex with using all sterile cell connector. And there is a chance like people tend to put an isolator which is not required. So
Okay.
that's the reason. So CAR-T is something which is different. You need to understand the process and accordingly you need to act rather than implementing the old systems. Unnecessary, you are burdening your own process and all those
Mm-hmm.
stuffs.
Okay. Understood. Thank you, Ramakanth. So, move on to the next question. So, how do global players approach scaling from clinical to commercial CAR-T manufacturing?
So global players, see, it's again how at what scale they are thinking on. For example, some of the products it all developed in a lot smaller scales. Some are developed in a large scale. So when you're
Mm-hmm.
talking about global products, mainly on most of the companies currently are developing in the clinical trial stage, and some are in the market. And the clinical stage is the where your data is very important in the clinical trial. So during this stage, the process will is almost developed and it's ready to kind of manufacturing. So also there is a lot of new technologies coming up, how quick you can manufacture the product. For example, some are
Yeah.
like 14 days, some are like 7 days, some are a rapid process. So
Yes.
different companies are approaching in different way how fast you can uh, give the patient back the modified T cells, because
Yeah.
longer the T cells are away from the body, it's not good. So how fast you can give the modified T cells to the patient.
Patient.
So
Mm-hmm.
in this case, there are many companies. This seller is those are all like they are manufacturing in a small box. Now everything happens within that cell kind of thing. So from the patient apheresis to till transduction, everything and giving back to the patient. So lot of technologies are there. I don't want to specify any company names. Not much into that, but these are all the approaches currently, Prashant.
Okay. Okay, Ramakanth. And on the same note, like, how does a closed system technology like a Prodigy uh, change facility requirements compared to a open system of CAR-T
Mhm.
manufacturing?
So again, whenever the word comes open, right, there will be a lot of questions. How you are
Yeah.
handling that open procedures,
Yeah.
either in an again here again you can say we can come again back to the isolator. So whenever you are having an open process, right, prefer to have
Mm-hmm.
an isolators, or
Yeah.
again background B with A grade labs, all those things will come up. But CAR-T, I feel like there are not much open process. Most of the systems currently are in closed process using sterile to sterile connectors. Even filling happens as if the bags, the expansion is done and it is transferred to the other bag in a closed process. So that is the best way, Prashant. Closed process is the best way, and most of the people are doing in this. Even G-Rex is giving all single-use transfer
Okay.
systems. Cocoon is a
Mm-hmm.
closed
Mm-hmm.
system. Prodigy,
Mm-hmm.
all those are all are closed systems. So closed system is preferred. So this also
Mm-hmm.
helps you. Either you want to build it in hospitals. So hospitals, you can directly manufacture in the hospital with a minimum space. If it is a complete
Mm-hmm.
closed system, you can go with C grade or even D grade based on your risk assessments.
Okay.
But if you are using like PL70 bags,
Mm-hmm.
certain kind of open systems, for sure you
Mm-hmm.
need to have a lot of control
Okay.
on the external
Okay.
environment.
Okay. Okay. And the next question is very important, and this is a challenge even we quite often face when we are, design consulting for a CAR-T project. Like, what is the ideal classification for a CAR-T facility? It's considering the closed system in place. So, there are lots of ifs and buts and ambiguity with the guidelines, and also what we have seen based on the regulatory audits. Certain auditors are okay with C, certain auditors compel for grade B, and there is always a debate even with the technology provider that because it's a closed system, it's okay even if you do in grade C, just do it in a biosafety or even a suspended lab is enough. So what is that, ideal classification for the core area people should be considering, Ramakant?
Uh, so Prashant, so normally these guidelines, most of the guidelines are either for an parental kind of things, and
Yeah.
later we try to put those things into CAR-T facilities. So
Yeah.
that might not be a good approach, what I feel,
Okay.
because see, in parenterals or in any MABS facility, the filling might have, it's all obviously happens in an
Hmm.
open systems, and even the formulation, you are adding the material and all those stuffs. But in CAR-T, right, especially if it is an autologous,
Hmm.
it's a patient-based therapy, right?
Yeah.
So you are doing everything in a closed system. You are adding a viral
Hmm.
vector. You are adding the you are doing apheresis. Everything is in closed system. If everything is in closed system,
Hmm.
uh, external environment doesn't matters.
Okay.
It's not a critical external parameter other than the temperature and humidity.
Humidity.
Um,
Yes.
but you are uh, at least you are maintaining the sterility within the closed systems. If you are able to
Yes.
maintain the within the closed system, and you are able to justify those things with proper risk assessments and all those things, I think any of the auditors should uh, be happy on that.
Hmm.
But as such, that's what I need to tell you again. Normally, we go with the past philosophies, always filling should happen in A grade.
Grade B. Yeah. A grade
Okay.
filling. Sorry. Yeah.
So, yes,
Surrounded by
agreed.
grade B.
A should be, filling should happen in A grade for an open systems, because,
Hmm.
but if it is in closed system, irrespective, because everything is happening in closed
Hmm.
conditions, there is no, the product is never exposed to outer environment, then what is the need of A grade? What is the need of isolator? So that's what it mainly, the thing is how your process is driven,
Hmm.
how your products are developed, and how you can easily transfer the product to the manufacturing. And based on
Okay.
that, you can decide your surrounding environment.
Okay. Okay. Fine, Ramakant. But what is that still you suggest? Like, should we go with bare minimum grade C and grade B is good to have?
Uh, C is good to have, but if it is
Okay.
a closed system, people are going with D also in some countries.
Oh, okay. Okay. And also, I think people, should keep this in mind because also grade B comes with, higher costs and different AHUs. So considering that, so C is something even what we have seen regulators accepting it, but there have been certain tricky situations wherein some regulators from the same regulatory body, they might ask for grade B. So this was a challenge for many of our clients.
Correct.
So, as you are telling, this cannot be compared to the conventional system of fill finish area. So, based on the other risk assessments, and at least it is good to have grade C surrounding the main systems, right?
Yes.
Okay. Okay. Fine, Ramakant. I'll also take a question from the audience, Mr. Ganesh Bhatt. So the question here is it advisable for startups or companies in early stage to plan ahead for risk assessments and scale up? If yes, how early should they consult?
So risk assessment to startup, you mean to say like I don't know whether you have a product already in your mind, or it's like you have already product and maybe you are in the
Hmm.
early stage. Yes, it's
Okay.
very important going with the risk assessment, because see, uh, in the early stage you will be freezing your uh, process. So on what way you are going up? For example, if you are going with an G-Rex
Hmm.
technology, then you need to take
Hmm.
care of certain kind of you need to observe certain kind of risks on all those things. If you're going with cocoon, there are certain risks. So your product also react according to uh, different media, like
Hmm.
how it is interacting with cocoon, or how it is interacting with prodigy, how it is interacting with G-Rex. So risk assessment, yes, it's always recommended
Hmm.
during the early stage, and at least you can predict, or you can jot on certain options like how can be my facility. So
Okay.
what might be my facility? For example, I can go with C grade if I go with
Hmm.
cocoon. No, my technology is in PL70 bags or some kind of permeable bags. Then you need to
Hmm.
think about what kind of facilities and how cost effective and all those things can be easily done.
Okay. Okay. Fine. Hope, Mr. Ganesh, that answers your question. And moving on to the next question related to the design aspect. So, what are the key HVAC considerations for contamination control and biosafety?
Again, Prashant, so here the main thing is the viral vector. These viral vectors are normally
Yeah.
coming from the, I can say, the part of the HIV virus.
Mm-hmm.
Uh, so if that's the only, I feel we need to take care of controlling and on the contamination controls mainly. So better to have a dedicated HUs for the manufacturing side, not mixing up with the other areas. That's
Okay.
my first preference. And also having biosafety cabinets when you're actually adding viral vector to the product during your transduction phases, or when you're transferring some medias, even though you are doing it um, in close process using sterile to sterile connectors, there might be chance like sometimes there might be a breakdowns kind of thing. So at that time, it's at least the minimum biosafety cabinet is preferred, but not on the, it doesn't controls, no, I can say that sterility anyhow, if it is breakdown, obviously the sterility is gone off. But on the safety side, biosafety cabinets are required.
Fine. Fine. So, on that contest, the next question is continuation of that. Then, how should the QC labs be planned? What critical factors you need to keep in mind, uh, especially regarding adjacency to manufacturing?
So QC labs, see, it's all depends on what capacity you are manufacturing.
Okay.
So you can even outsource the QC labs.
Mm-hmm.
Microbiology can be outsourced if at all. You're planning like, for example, if you're planning like 4 to 5 batches,
Mm-hmm.
each batch will take minimum 2 weeks normally. If it is a rapid process, depending on the technology. If you are going with one model within the lab, better to outsource the QC. That's what my preference, rather than you spending much money on the QC, especially on the sterility testing. You need to have all
Yeah.
the labs, different labs. Better to outsource. But if you are planning for like 80, 90 batches a
Mm-hmm.
month,
Mm-hmm.
then you can have adjacent QC micro testing and all those stuffs. And also
Okay.
here, very important thing is testing and releasing is also an important part. You
Yeah.
cannot wait 14 days for releasing a test. So nowadays, like this rapid sterility testing kits are also there. So I think within 3 or 4 days, I think you might be expert in that.
Mm.
Three
Yeah.
or 4 days, you will be getting the test result.
Yes.
So these kind of technologies helps to release the product at the faster rate.
Okay. Okay.
So
Fine, Ramakanth. But one question here was like keeping the time factor. So, so though you outsource the microbiology testing to outside third party, but it is better even they have the rapid system so that you get the data in real time, so that you can release the batches and give it to the patient. Correct?
yes,
Okay. Okay. Because that is very important, because many people, when they tell that they are ready to outsource, but they should always keep in mind that the outsourcing lab also should be again the regulatory body approved, and they should also have the rapid system so that they can support you with the data in real time. That is a
yes,
very, very important thing, because you have put
yes,
so much of effort, develop the entire batch for 9 days or 14 days or whatever days, but if you don't get the data in real time, in matter of 5 6 hours, whether it is endotoxin, sterility, and even the flow data, then
hmm,
it becomes because every hour the cells will start losing its potency. So it's very
correct,
important that the batch is administered at the earliest. Okay.
correct, correct. You are right.
Yeah. Thank you, Ramakant. So, and also, like, uh, the next one is also very important. This is for most of our audiences who are also designing the initial setup of the CAR-T. Like, how should material and personal flow be designed to avoid cross-contamination?
So it's a normal flow, Prashant. There is nothing special in for CAR-T. It should be unidirectional as you
Hmm.
follow for the other facilities, and
Okay.
a product should all the raw materials should come from one side, and the waste should go on the other side. There is no much specialty for CAR-T. What I
Mhm.
mean to say, it's a regular process. You can follow the fill finish man material flow or in DSDP man material flow unidirectional with
Mhm.
proper uh, double wrapping and all those things, having certain pass boxes, air locks and all those things. So I don't want to much put on that because everyone knows this. It's a regular man material flow for
Okay.
an industrial thing.
Okay. Okay. So the most important thing is the unidirectional thing, designing of the pass box in the right places as and when required, and overall handling of the products or the reagents. So that is
Yes,
what you are suggesting, Ramakant.
yes.
Okay. Okay. Now, with all the technology advancement and everything, so what do you think, like, how important is automation versus manual intervention in CAR-T manufacturing?
Automation and manual intervention. Uh, see, uh, it's important because see, it starts with the patient entering into the hospital. I can say like how important is automation is like the logistic part is completely should be properly well managed.
Mm-hmm.
So when the patient joins hospital, he get registered. Once he get registered, the diagnosis should happen, and it should be find the suitable product from different companies. Maybe once that is done, then the FRSs and the doctor will do the FRSs of the patient, and then the samples are transported. So automation, I can say like it's very important, and also the data management is very important in this case because it's a patient-based therapy, especially in
Yeah.
autologous. We didn't talk
Yeah.
about allogenic, right?
Allogenic, yes.
So especially here it's a patient base, so the data management track and trace is very important when you are actually working on the autologous. So automation plays an important role in this.
Okay. Okay. Fine, Ramakant. And see, keeping that in mind again, what role do digital systems, so all the other systems are which are also part of this CAR-T manufacturing, uh, play in CAR-T facility readiness, especially keeping the 21 CFR in mind. So there is always a notion, discussion, debate at site that all the other, uh, it's usually an industry practice that most of the instruments or equipments which generate data should be mandatorily 21 CFR. But is it a mandate globally that everything or anything you are going to use should be 21 CFR?
So it's always important to have a 21 CFR because whatever you are generating is compliant. You are
Yeah.
unable to manipulate any of the data. That makes
Hmm.
you uh, uh, the patient, or the
Patient.
customer, you trust them because
Yeah.
every data is not manipulated. Whatever you
Hmm.
do, the changes, it's all recorded. So it's always important to have a 21 CFR data without any, all data recording should happen. At the
Okay.
same time, uh, you know, the tracking of the patient, the barcodes which are generated for the patient, it sticks onto the bag, and then that bag
Yeah.
transports to the um, manufacturing site, and they scan it, and you are getting the right patient, and then a lot of labels are generated, and then that goes to the, so this tracking each and every step with respect to the patient, and again
Hmm.
the same should go back to the patient, to the same patient. That's also
Yeah.
important. It cannot be given
Yeah.
to other patient. There is a chance because
Hmm.
blood looks same red for everyone.
Yeah. Yeah.
So that's the reason. So tracking the product is very important.
Okay. Okay. And we also faced multiple, CAR-T facility audits and part of many, audit observation justification. So what has been your experience, especially the regulatory journey of CAR-T facility approvals in India compared to globally?
So that's what, as I was telling previously, you have an auditor who knows about microbiology, for example.
Hmm.
So he can easily understand the way you are doing in microbiology, for example. And if I give a physics guy as an auditor and you need to explain about microbiology, it's difficult.
Hmm.
So same thing. So in other countries, it's easy because the technology and the transparency and the education to the regulatory bodies is very quick.
Hmm.
But I can say, in India, maybe I can say that CAR-T is new, and you
Yeah.
need to educate the auditors, especially
Yeah.
SEDESCO
Mm.
or RCGM, all those
RCGMS,
teams.
IBSC, RCGM, all these committees, yes.
Yes, we are not against anything, but educating makes them understand
Hmm.
what actually
Yeah.
it is. So you
Hmm.
cannot put some other regulations of other
Hmm.
products into CAR-T. At the
Yeah.
same time, you cannot put CAR-T regulations into other. So you should have a CAR-T dedicated regulations.
Okay.
So that will help for easy fly in a more compliance and high quality products, and
Okay.
also cost efficient.
Yeah. Yeah. And on that note, there is a one important question which even I had, and one of the audiences also asked. Again, it is BG. Currently, all player providing solutions in CGT space are big global giants, which increases the cost for patients, which is true. Do we see India developing its own ecosystem in near future, or have you come across any player who is already working on this? Ramakant.
So India is actually doing good on CAR-T space. I think there are 3, 4 companies. Everyone knows this. I think uh, Immunil, Immunoact, they are doing very good. I have seen their presentations in US,
Okay.
and they are doing a very good work and at a very cost effective
Okay.
compared to US, UK, or any other countries.
Mm-hmm.
Uh, Again, how they are doing, it's all depends on what methodology, what technologies they're using it. And yes, India is a good market for CAR-T. And I was also read like when you were young, Prashant, do you know how many diabetic patients you know when you were young?
No, it
Well,
was very less. Yes,
now how many diabetic patients are there?
it's very common.
Common. So maybe
Yeah.
2030, cancer also becomes common
Okay.
because uh, uh, it's I can say many people are getting stressed, or a lot of mutations are happening, many new things are coming up, a lot
Mm-hmm.
of life, all those things. So obviously you are giving more stress to your own cells that leads to
Mm-hmm.
cancer. Maybe I don't know. So that's the reason by it's always India is growing, and you will get lot of opportunities in India. And yes, India can be the right place for an economic and I can say quality product for karting.
Okay. Okay. And we were also discussing about autologous and allogenic. So, can you please explain our audience what is the key difference between these two and what are the developments in the allogenic space? Uh, whatever the updates you have.
So autologous is patient to patient, means
Yeah,
the patient sample is taken, you modify the T cells and grow the T cells and give it back to the same patient.
that is what I
Allogen
believe most of the systems are currently. All
currently,
the technologies are depending on autologous.
correct.
Yeah.
Allogenic is you are taking T cells from a healthy donor,
Hmm.
and uh, you modify and give it to multiple patients.
Okay.
So allogenic is, I can say, something like a DS process. You will have a small bioreactor kind
Hmm.
of thing, and you have a healthy patient, and you need to grow, and again you multiply and make it in different vials or different bags and give it to multiple patients. So this is the key difference in general terms. Um,
Hmm.
See, both are different process, but I can say like viral vectors, transduction, expansion, everything is same, but allogenic has an you do it in a bioreactor kind of thing, small bioreactor.
Okay.
That's the key difference, Prashant. But
But what is the success
how
factor in allogenic? Have you come across any
yeah,
latest
there are a lot
research
of
or studies?
research happening on uh, brain tumors and all those things.
Hmm.
Uh, People are working on that, and also there is also new technology like you grow your own T cells within your body rather than you grow it outside. So
Oh,
this
okay.
kind of technologies are coming up,
Hmm.
but we don't know like when it actually flies. But for sure, maybe in coming 5 to 10 years, many technologies will be in the right path and serve a lot of cancer patients, I feel. And also I heard some of the new paper which I read was they isolate the T cells, and they make you do the gym, right, to strengthen your body. Similar
Yeah.
way they strengthen the T cells and give it back to you.
Yeah. True.
Even though you don't have cancer, you
Yeah.
just strengthen it and give it back to the human so that they
Hmm.
help to fight unnecessary stuffs, whatever the
Okay.
invades.
So yeah,
So,
probably something is in early stage or even something is coming up. It is just like having a vaccine for cancer, something like that.
yes, something
Okay.
natural. T cells, your own T cells will help. When you're growing, your
Okay.
T cells also will be aged, right?
Okay. Yeah. True. True, Ramakant. And the next question is like, okay, when we do design the facility, you do have a facility, but considering like probably you have one technology and one or two systems now, but tomorrow you might end up having 8 to 10 systems based on the needs. So when you are considering that, what design factors people should keep in mind so that the near future expansion also is taken care.
So here again, sorry, hello,
Yeah, Ramakant, we can hear you.
second. So there are different types of facility designs, Prashant. You were talking about how easily you can expand, right? So it's all depends how you are planned for the future, like 5-10 years. Normally, it can be a ballroom concept, wherein which all the activity happens in the same room. Okay.
Yeah. Mm.
In some other case, we segregate that ballroom concept into like 3-4 rooms,
Yeah.
and you do the cell separation in one room, and uh, activation or transduction in one room, and expansion and finally formulating and filling in one room. So this kind of approaches you can go based on your markets. If you are uh, having a strong belief that there is a sure, like I need to increase my batches,
Mhm.
so you can have multiple ballrooms. Maybe you go with one ballroom concept currently and add on
Mm.
multiple ballrooms,
Yeah.
or you initially itself design with multiple rooms, and
Mm.
you can increase the respective unit operations in each room.
Yeah.
So either this process
No,
you can up.
why this question is important, Ramakant? Like recently we came across a client wherein they had designed the initial concept with having just one or two systems, but suddenly they got the success and now they want to go more aggressively. The room is not having the sufficient space wherein they easily need another 6 to 8 systems. So now the question here is about the GMP readiness. So if you try to touch that room and expand it, then again
Mhm.
it attracts for more audits and everything. Whereas already you have sufficient space and you, as you rightly mentioned, the adjacent ballrooms, so you can keep expanding those. Still there will be a visit, but probably not so much of again it becomes like a small brownfield project wherein you have to dismantle the panels, do the entire thing. So that is where this question was repeatedly asked to me also, like what is the main factor, considering the future expansion also, how we need to keep the core area ready so that there will be minimal GMP impact.
Yes, that's what I said. So based on the segregations, you have already the existing facility running. You need to properly segregate, and you need to have a space for the future expansion, plan it properly, and make sure you replicate the same design rather than having multiple designs. In the first phase you do it one design, and the second phase, third phase you do it in a different way. So better to have a replica of A, B, C, D in the same way, and so it's easy to you can save a lot of cost, and your process is same and all those things. But whatever you do, do it first time the right way.
Yeah, 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 true. And with this, I have one main question. After that, this floor is open for the audience. And also, audience, after this Q&A session, we have one more important announcement. We have also Mr. Arun Singhal with us, who is from Biotech stage. So we'll be also introducing Arun Singhal here, and he will be also mentioning about upcoming event which is happening in Hyderabad on April 23rd, 22nd, and 23rd. So please be there. Okay. So what advice would you give to organizations planning their first CAR-T facility, Ramadan?
So first thing is whoever is willing to start their CAR-T facilities, make sure your process is designed properly, means in your R&D stage you have a very strong clinical data, uh, because this clinical data reports will help you how your product is reacting to different individual patients. Even though the
Mm-hmm.
product is same, you know, the T cells are different for each and every humans, right? So clinical data plays a key role. And third thing is how robust is your uh, process.
Mm-hmm.
So how quickly you can manufacture or come into the manufacturing stage. So these three will be the key things to be considered if you are actually planning for CAR-T facilities. Obviously, you need to have the right talent for sure,
Yeah.
and uh, you know you should also analyze the market, how you are planning, whether you're planning tying up with hospitals or you're planning independently and try to get the patients, or you are planning for a medical tourism. So these are all multiple options how we can collaborate. But yes, for sure, I think future CAR-T will be a key player in our current pharmaceutical industry. I think MABs,
Mm-hmm.
ADCs, CAR-Ts,
Yeah.
these are all like key players maybe in the down the line 5 to 10 years.
Okay. Okay. Thank you, Ramakant. And now, over to you, audience. Like, was a great discussion, Ramakant, and thanks to all the audiences for being part of this great Q&A session. And special thanks to Ramakant for being part of this. I know it's very late evening for you, and but still, you're part of this and gave us all the GAN which we needed
Mm.
regarding CAR-T, especially the facility designing and the future of CAR-T.